Hey everybody! Welcome back to the Lab 207 webcast. My name is Mr. Nasiri, and I'll be joining you today. We're going to change it up a little bit. Mr. Kite ended up having to take care of his baby, so I'll be filling in. I'll be doing things a little bit differently. First off, I'll be adding a little background music. Let's go ahead and get started. Today's topic is going to be about extracting DNA and using it effectively. Couple of objectives for you real quick, just jot them down. First off, students will be able to describe how scientists would recover DNA from blood samples. Next, students will be able to understand how scientists use PCR to amplify recovered DNA fragments. And finally, students will be able to explain how scientists use genome sequencing and viral genome databases to construct a phylogenetic tree that could be used to show the relationship between old and new strands of DNA. That was a mouthful, but let's go ahead and keep going. Starting off with extracting DNA from a blood source, as you can see on my chart to the left here, there are many ways to extract DNA from a blood sample, but the best method for HIV is a solution-based DNA extraction method. More specifically, the organic solvent, catotropes method. In this method, cell lysis is undertaken by adding a detergent, catotrope containing solution. Wow, words are hard. Followed by an activation of DNases and RNases, usually through the use of organic solutions. Then, purification of DNA and removal of RNA, lipids, and proteins. And lastly, resuspension of extracted nucleic acid. The next topic we're going to talk about is to describe how scientists would use PCR, polymerase chain reaction, to amplify the recovered DNA fragments. In the PCR reaction, a three-step cycle brings about a chain reaction that produces an exponentially growing population of identical DNA molecules. So 3 steps. The first step is denaturation, which is the reaction is heated to denature or separate the DNA strands. Next is annealing. What you're going to do is cool down the DNA strands to allow hybridization of short single-stranded DNA primers complementary to sequences on opposite strands at each end of the target segment. The last step of the process is extension. A DNA polymerase extends the primers in a 5', 3' direction. PCR is speedy and very specific. Only minuscule amounts of DNA need to be present in the starting material. Alright everybody, we're at the last topic of the day. I know it's been a long video, so I'll try to get through this a little quickly. The last topic is to explain how scientists use genome sequencing and viral genome databases to construct a phylogenetic tree that could be used to show the relationship between the old and new strains of HIV. Now, genome sequencing allows for scientists to know the different base pairs that make up the HIV virus. These sequences can then be used to calculate genetic distance. These are one of the characteristics that can be used to measure how far apart 2 different strands of HIV would fall on a phylogenetic tree. They can then place the different genome sequences in a database where they are stored and kept. In the future, any other HIV strains could be compared to the ones in the database. This would be a character-based approach. With this database, scientists are able to find the strand of HIV that is most distinct and make that the outgroup. From there, all the other strains can be placed on the tree according to their similarities and differences. An indicator of variation could be a new base pair combination being introduced that wasn't in the previous strains. Well, hope everything went alright. Hope you got everything, understood the concepts. If you didn't get anything, skipped over something, just go back, rewind, look over it again. That would be the end of this webcast. See you again. My name is Mr. Nasiri. And thanks for joining us.